Okay, here goes a lesson on enzymes. It's one of two, so if you think to yourself, I think I remember learning more about enzymes, there's a reason for that. So before we get started and go into more detail, what are enzymes? Well, enzymes are proteins. So that is important to keep in mind. Anything that applies to proteins that you've learned, from the monomer all the way to the different levels of protein structure, are going to be true of enzymes as well. And they catalyze chemical reactions. Don't worry, I'll go over catalysis more on another slide. All of the names of enzymes end in ase, so there are things like sucrase, there's lactase. Those you may have heard before. We will talk later about kinases. And we might do a lab on either catalase or lactase. And these reactions can be either synthesis reactions, like this one shown here, or decomposition reactions. I'm going to put something in your notes where you need to identify another term for synthesis as well as decomposition based on what we learned about with biomolecules and with monomers and polymers. So, some important terms to remember. The substrate, which is the orange molecule here, before it's been broken down, is a reactant which binds to the enzyme and will be changed by the enzyme. Because other things can bind to enzymes, but they're not changed by it, and so they won't be substrates. The enzyme substrate complex is a temporary bound association between the substrate and the enzyme. Pretty straightforward in the name. The product is going to be what is produced at the end. So here, this is actually a sucrase reaction. So here our substrate is actually sucrose, a disaccharide, and then what you get at the end are two monomers. You get glucose and you get fructose. So those are the products of this reaction. And then finally, the active site is the area where an enzyme binds to a substrate, and it's also the catalytic site where you're going to actually perform the reaction. So there's the active site of this enzyme. So I've talked about catalysis or catalysts a couple of times. What do? They do a couple of things. They lower activation energy and they speed up chemical reactions. And they do this by one of a number of methods. They can, if they need to, if they're breaking down, especially a larger molecule, they can basically create strain or induce strain in a bond where they kind of stretch that bond and create what is called an unstable transition state. So a transition state is kind of part of the way between substrate and product, but it's not stable. So that's why it will very quickly turn for a substrate into a product. Another thing is if you're doing a synthesis reaction, you can actually have the enzyme put these two substrates into a position where they can bond more easily. And then you can also have R groups directly involved in this reaction and add chemical groups onto something. So we mentioned activation energy. Activation energy is basically just the energy you need to start a reaction. So you have to input an energy into any chemical reaction for it to take place. Enzymes lower that activation energy. So I'm going to add that and I'm going to leave a space in your notes for it. So enzymes lower the activation energy. And if we look at this reaction that you see here, let me go back. If this had an enzyme helping the reaction happen, then the activation energy might be something like this. So that difference in activation energy, that is what the enzyme does. So it makes it easier for this reaction to happen. It takes less energy for this reaction to happen, which means it's going to happen faster or more often. So there are a few things to know about the enzyme substrate complex. Basically, the shape and the charge of the substrate have to match with the active site. They have to be compatible in order for a reaction to happen. And then the enzyme and substrate, when they're together, they're held together by either hydrogen bonding, there may be some electric attraction between differing charges, or there may be a temporary covalent bond formed. So which of these two substrates, the orange one or the green one would fit into this enzyme? Well, it's the orange one because its shape matches the active site. There are a couple of different models of how enzymes and substrates bind together. One of them, which you probably learned your freshman year, is lock and key model, where the substrate fits into the active site and they match perfectly, which is why the enzyme is able to act on that substrate. The other type of model, which may be a little more accurate molecularly speaking, is the induced fit, where the actual binding of the substrate makes the active site change in such a way that it can help catalyze that chemical reaction. Now, enzymes, and we're going to talk in the next lesson about things that affect enzyme activity, but you can have some other molecules involved. So you can have activators like cofactors. These are non-protein, small things like ions or inorganic compounds, maybe magnesium or iron, that bind to enzymes and help them function properly. There are also coenzymes, which are also non-protein, but are organic molecules. And these guys can bind temporarily or permanently to an enzyme near its active site, and they can participate in reactions with more than one enzyme. They're not enzyme specific. So you don't need a specific coenzyme or cofactor for every single enzyme that you have in your body. And a lot of these are vitamins, which helps explain why, you have these daily recommended allowance of various vitamins, especially the ones that we can't produce with our body because they're needed to help our enzymes function properly. So here we see in a diagram, we see an enzyme. This is the protein part right here. Trying to get the laser pointer to show up. There we go. So this is the protein part right here. Here is a non-protein cofactor. And notice that when it sits in the active site of this enzyme, it makes the active site match the shape of the substrate, which helps that reaction to happen. And some of this regulation happens at the active site, like in the previous diagram, but some of it can also happen at a different site. This is called allosteric regulation, meaning it happens at a different spot. So it's not going to directly interact with the substrate or the active site. It'll be somewhere else. And you've got either activators or you've got inhibitors, which hopefully their names tell you what they do. Activators will help the function of the enzyme, maybe make it more active or make it possible for it to bind to its substrate, and inhibitors will block enzyme function. So it could be something as simple as they change the shape of the active site so that it's less likely a substrate can bind. They may induce, changes in charge, something that would prevent a substrate from binding, just like an activator would potentially change an active site so that a substrate can now bond. So that is it for this lesson. We're going to talk a lot more about enzymes in our next lesson.